Dr. Foster. He is an associate professor in the Department of Biological Sciences and the Pathogen and Microbiome Institute at the Northern Arizona University, where he is standing out and giving this presentation in the parking lot. His interests are broad within ecology and evolutionary biology, but he largely works with birds, bats, and livestock, and often their pathogens. He has collaborators and projects throughout the world, particularly for infectious disease work. And of course, coming out of or coming through a pandemic, we are all now very much, probably hopefully more interested in infectious diseases and how they spread. So I think this is a very timely conversation. So with that being said, Dr. Foster, I'm going to hand it over to you. Thanks for coming, and you are welcome to take over on the screen share.
Okay, hold on. Disappear 1 second, please.
Yep, take your time. There you go.
All right, you see it?
Yep.
Okay, great. All right, thanks, Adam, for the introduction, and thanks to the Vice President for Research Office at NAU for inviting me here to speak. And so, yeah, so indeed, kind of at the tail end of a pandemic, or hopefully at the tail end of a pandemic, it certainly is timely to be talking about infectious diseases. And I think we can really relate to it in particularly how fast an infectious disease can spread globally. And so I too will be talking about infectious diseases and how they spread globally, but over millennia and not over days or years. And so very similar story, but over a longer time period. And so I was actually recently in Italy at a brucellosis conference, and it turns out this does not, the cow jumping over the moon does not translate into Italian or most European languages, and so it fell short. But this one didn't fall short, and so no one has seen a goat jumping over the moon. So today I'll be talking about two major livestock diseases in both cattle and in goats, and we'll get to that in a second. And so for those of you who are a bit older, this was a pretty prominent campaign, kind of got milk with a bunch of celebrities, have milk mustaches and out there. And I am not funded by the dairy board, and they would probably shoot me if they saw this. And so got Brucella. And so actually milk is the #1 way people get brucellosis worldwide. And we'll be talking about that later, but not in the US. And so we don't have any Brucella in our milk, but certainly it is one of the most important pathogens worldwide. And so I'll be talking a lot about Brucella today or tonight. And so this story, we think, begins on the island of Malta. So I've circled Malta kind of down below Sicily here on this map in the late 1880s, where a military officer and surgeon, Sir David Bruce, was stationed there. And he realized that a bunch of his soldiers were getting sick on this island. And so they ultimately determined, and it took several years, but they ultimately determined that goats and contaminated milk with Brucella were making the soldiers sick. And so it was a long kind of detective story to try to figure out the source. And lots of different ideas, but they're able to culture bacteria out of milk and ultimately identify that that goats were responsible. And so, but the real story actually goes back way further than the 1880s. And so this is a study that that came out in 2009 by D'Anastasio, and the conclusion was that there's certainly a suggestion if you look at fossilized bones, and this case is from Australopithecus in a greater than 2 million years old. There are lesions on the vertebrae of this hominid that are consistent with brucellosis. I can't tell myself, but certainly experts suggest that this early our ancestor may have had brucellosis. And so, but certainly by the Bronze Age and then by the Romans, we have more and more fossils, and there are definitely lesions that were caused by brucellosis. And so if you look at different vertebrae and other bones of people from this time, we can find definite evidence of that they were infected with brucellosis. So we know it goes back and infects humans for, a long period of time, and potentially even from a cheese that was preserved and had actually a consistency or an appearance that it had brucellosis in it, particular bacterial structures in the cheese. Who knows, but certainly suggestive. And then even more recently, and this is a pretty amazing study where brucellosis causes, it's a disease that people get primarily, as I mentioned, from milk and dairy products from infected animals. And when people get infected, they can get calcified nodules, throughout their body, typically kind of near their spine. And if a group from the UK went into drilled into these ossified nodules and was able to extract DNA from skeletons from the 1300s. And we know about this most prominently, say, within Yersinia pestis that causes plague. People can go back to the 5th and 6th century and find plague DNA inside teeth, for example. But in this case, they're able to sequence an entire genome of Brucella from this skeleton. And I'll show you exactly kind of where it gets placed in a phylogenetic tree. So it turns out what we can find from the 1300s is still there and present. The same lineage is still there and present in the same area of the world. So we have, I hope I've kind of convinced you that humans have been infected with brucellosis for at least thousands of years and potentially millions of years. But we know that this is a zoonotic disease. So much like SARS-CoV-2, that we got it from animals, likely bats. In this case, we know that the pathogen Brucella is coming from animals. And so these are the kind of major livestock that we think about that are infected with different species of Brucella. And so let's go back and think about the origins of livestock. And so interestingly, and so the kind of keynote speaker coming from Egypt and talking about kind of Egyptian ancient history, certainly the connections are apparent where the origins of civilization are, just down the road here in the Fertile Crescent, and the kind of intersection of Turkey, Iraq, Syria, and Iran. And so in this time, and so roughly 10 to 11,000 years ago are the origins of most of the livestock that we think about today, and in fact have in our agriculture. And so the origin of the 4 major groups of livestock are all in this roughly the same area. And so, but we've spread our livestock from there globally. And so there's 2 reasons for this. One is, for some of them, we depend on dairy, and dairy isn't the whole story, but we depend on dairy and we depend on them for meat. And in some cases, we depend on them for wool and fiber. And so we are dependent on animals, and we've spread those animals from the Fertile Crescent and potentially a couple other places in the globe around the world. And I'll show you lots of pictures about how we've done that. And so this is this is one way that we're transporting them potentially across water. And so we now have cattle on 6 continents. In fact, cattle are just about everywhere in the world except kind of the Arctic and our extreme deserts and certain mountaintops. And so we've spread cattle everywhere. And so this is a worldwide range of cattle. And so we can see that, there's particular parts of the world that more cattle are present than others, particularly India shows up, parts of sub-Saharan Africa, and parts of North and South America are especially prominent. But pretty much everywhere we've been, or nearly everywhere we've been, we brought our cattle. And we've also done the same thing with our goats and sheep. So here's a worldwide range of goats. And so I'll have you pay particular attention to sub-Saharan Africa. And so we'll come back to this, but take a look. Goats are everywhere, but they're unevenly distributed. And sub-Saharan Africa actually has a lot of goats. And this is the worldwide range of sheep. And so a bit of a different distribution, but we know that sheep have been spread for both dairy as well as for wool and meat. And this is the way that I like to think that Brucella, or at least some goats, can get around. And it turns out wherever we bring our animals, we bring their diseases. And so Brucella is hitchhiking along everywhere we're bringing our animals. And so Brucella abortus, and so which is primarily in cattle, and Brucella melitensis, which is primarily in goats and sheep, spread along with their hosts to the 6 continents. And this is actually a fairly common story. We can look at different bacteria, for example. And so in the top left, that's Helicobacter pylori. And so there's great studies about how it causes ulcers in people and how humans have spread Helicobacter pylori globally as we have kind of colonized the world. And so people bring Helicobacter in their guts, and we can look at the bacteria and the genetics of the bacteria to look at that spread. And then in the top right, we have Mycobacterium tuberculosis. And so again, another bacteria that we can track its movement genetically. We can look at the genomes of this bacteria and see where it has been and likely how it's kind of gotten to those different places. And then finally, and kind of maybe one of the stars of the show, in fact, prominent researcher at NAU named Dave Wagner, who works on Yersinia pestis, which has caused the plague. And so think bubonic plague, but it happens even today. And so you can find Yersinia pestis within the Flagstaff city limits if you know what you're looking for. But researchers can look at the genomes of Yersinia pestis and be able to use that to infer how Yersinia pestis has spread globally. And so we can use DNA to look across time and space. Hopefully everyone gets the Doctor Who TARDIS reference. And we can sequence entire genomes to do that. So we crack open the cells, look at the DNA that's in the bacteria, and then we can kind of make sense of all of that DNA and compare it against each other. And so and so one of the things that we do, and so this will be the most detailed slide I'll show of genetics today, so bear with me. And so we see 3 sequences from 3, say, bacteria in the top row that are in blue. And then we see 3 different sequences that are in green in the bottom 3 rows. And so they differ by just this one base. And so the first 3 have Gs, and the second group of 3 have As. And so we would infer, just based on this information, that all the blue ones are closely related to each other, and in fact identical, at least here, and all the green ones are related to each other. And we do this across, not at a single site, we do this across thousands, if not millions of sites, and see which of them match up. And so when they match up, they're most likely related to each other. And when they don't match up, they're more likely to be distantly related. So we kind of piecewise go through the genome and figure out how all the different samples are related to each other based on the mutations that they have. And so how do we assess these relationships? So we use phylogenetic trees. And so this is actually a tree from one of Darwin's early sketches. And the main thing I'll just point out here is that on a phylogenetic tree, if you're on a branch tip here, say where you see the letter B, those 2 samples or 2 individuals or 2 taxa or whatever you want to call them are closely related to each other. And the further away you go down on the tree, say sample A, that's distantly related from sample B. And so B and C are relatively closely related to each other, and A is distantly related. And we do that just based on the similarity or dissimilarity in the genomes that we have. But back to the star of the show, Brucella is actually far more than just a livestock disease. I would argue it's one of the most successful pathogens globally. So it's in our livestock, as I mentioned. It's in our wildlife, and so lots of different wildlife. In fact, we're finding new species nearly every month. And so the more we look, the more we find it. And it's in our oceans, and so it's in marine mammals, a variety of marine mammals, and it's actually in a bunch of different marine fish as well. But yeah, so that's just my pitch for Brucella is super exciting and has conquered the world. But let's talk about how it's conquered the world in terms of 2 species. And so the first one I'll be talking about is Brucella melitensis. And so this is the one that infects primarily goats and sheep. And then we'll talk about the Brucella abortus that primarily infects cattle, both dairy cattle and beef cattle. And so, as I mentioned, genomics allows us to track the spread over of this pathogen over ancient and recent timescales. And so we actually did phylogenetic analysis. This is pretty early on and using genomes to understand bacteria, at least Brucella. And one of the main things that we found was that there were 3, this is a very simple tree, but it actually is still true today, or nearly true. And so you'll see that we were amazingly lucky in the samples that we picked when we created this tree. So you see 3 distinct lineages, and we'll talk about each of these lineages in more detail. But the main point I'm making here is that we have 3 obvious differences, 3 lineages that were identified in genomic sequencing. And so then I'm showing you a much more detailed analysis with a couple hundred genomes. And so in this case, it turns out that there are still 3 main lineages. We see the lineage here in red, a lineage in the middle of this in blue, and then finally a lineage in green. And so they do have names, and you'll figure out why in a second, but that top clade or top lineage is the Americas clade. The middle one, the East Mediterranean is blue. And then finally in green, we have the West Mediterranean clade or group. But the more we looked, the more we found kind of interesting things. Oh, I'll point out, I did show you that sample from the Sardinian skeleton. It actually falls out right here in this phylogenetic tree. And so interestingly, all of these samples here, you can actually see most of their kind of names, say ITA. These all came from Italy. And so, and then we have one right at the very bottom that came from Egypt. But the main point here is that what we see here currently, and so see here in the past couple of decades, samples of Brucella melitensis were actually there, or that lineage has been there for over 1,000 years. And so we know that, yeah, what we're seeing people infected with today are, it's been there for a relatively long amount of time. But I had an additional study where I had a collaborator collecting samples from camels in Oman. And he was collecting these samples, and camels, it turns out, get infected with Brucella melitensis as well, especially when they're in close contact with goats and sheep. And so in a lot of areas of the world, particularly parts of the Middle East and parts of Africa, yeah, goats, sheep, and camels are in close contact with each other. And so one thing that we noticed that we hadn't noticed before, and interestingly, no one else had noticed, is that there's a really, if you look closely, within the red group, you see actually two distinct branches. You see a lower group that the Omani camels are in, and then the upper group that, and an upper group. And so we looked more closely. And so we were really targeting and looking at these samples, and the kind of the punchline is that nearly all those samples in that second lower group that we saw here, where the Omani camels were, are part of this at least until then, unknown African clade or African lineage. So it turns out we have 4 distinct groups within Brucella melitensis. We have a group in the Americas. And so if you go to North and South America, you'll largely find samples from there. And interestingly, if you go to those same samples or same lineages are found primarily in Europe, in Spain and Portugal. And so if you know your history, you may already be making those connections right now. What connects North and South America to just the Iberian Peninsula of both Spain and Portugal? We have an African group as well. And again, if you know your history, why would Omani samples be connected to Africa? Part of that is we know that there's major trade routes, particularly Arab trade routes all along that area. And then finally, we have an East Mediterranean group that we'll talk about. It's kind of a mixed bag. And so the more we looked, the more we found, and we found this African lineage being found in lots of different places. And so we're able to sample it extensively. And back to this major part of the story, that Brucella melitensis in sub-Saharan Africa is nearly entirely this African group that we hadn't really even known existed until we really knew what we were looking for, and then developed particular approaches to look for it specifically. And so we were able to identify 4 main lineages of Brucella melitensis. And so the first of these is the West Mediterranean, that has been in the Italy region for a long period of time. East Mediterranean group, which actually spreads from the Middle East all the way to East Asia, all the way to Korea and eastern parts of China. We have an Americas group, which includes Spain and Portugal. And then finally, we have this African group, which also includes part of the Arabian Peninsula. And so here's how I believe it happened. First is that we had a spread from early domesticated animals to the Iberian Peninsula, and then we had a spread to North and South America. Nearly all of these samples from these regions are closely related to each other. And it turns out if you run a molecular clock on this and try to date the timing of when those samples likely emerged, it happens around 1500. Again, you kind of have to take these types of analyses with a grain of salt, but it certainly is suggestive that they were introduced at roughly this amount of time ago, and that Brucella came early on into the Americas. So the African lineage has a slightly different story, where it spread likely to the Arabian Peninsula and possibly kind of across the Sinai to Egypt, and then spread back and forth and stayed largely within Africa. We can see that those lineages are still there, although they're certainly undersampled. Then we have this West Mediterranean lineage, which spread from the Fertile Crescent to Italy, and interestingly, and then spread slightly through to Italian colonies in North Africa. But interestingly, despite the Romans, conquering much of the Western world, they didn't bring Brucella with them. And if they did bring it with them, they didn't bring this lineage with them. They potentially brought another lineage, which we're about to see. And so finally, the East Mediterranean lineage, which in some ways is kind of misnamed. It was only because it was first seen in the East Mediterranean, but it really spread from the Fertile Crescent, throughout the region, and then particularly spread throughout Asia. And this is one potential way that it spread. And so we know that we have brought our animals with us, and we bring our animal products with us. And certainly this, both by land and by sea, we have spread our animals kind of across the largest continent on the earth. And so, so for the first part of this, and so for Brucella melitensis, I hope I've convinced you that humans have moved Brucella melitensis along trade routes. It arrived in the New World early in the colonization of the New World. The spread from Italy was surprisingly limited, but it has been there for a long period of time. It just hasn't gotten out very often. Little is known about the African lineage. The little known African lineage is actually quite common, and part of us not recognizing it is that Africa is really undersampled in terms of infectious diseases. And so it's hard to work there. They're bigger problems than infected goats. And so it's difficult to get large numbers of samples from there. And it appears that it spread rapidly through Asia, and there aren't a lot of differences, at least within the Brucella melitensis within Asia. Kind of some unanswered questions is the timing and effect of goat domestication and sheep domestication on this species of Brucella. And there are wild goats all throughout these regions, and so their connection to this disease is unknown. And so ideally, I'd like to sample wild goats and try to figure out, is there any connection to this particular species of Brucella? Did they play a part in it or not? We know that, say, ibex in France can be infected with these kind of quote unquote modern day Brucella melitensis, but were they infected in the past by other lineages? We don't know yet. And so we can also look at Brucella abortus. And so, as I mentioned before, Brucella abortus is primarily in cattle. It affects a wide range of hosts, but it's primarily in cattle. And so this is just another way of looking at a phylogenetic tree. It's not rooted, but the key thing to see here is that on the left-hand side, we see kind of all of these short branches, and that indicates that all these samples, in fact, there are hundreds of samples in there, are all super closely related to each other. And it turns out these are samples from most of the world. Most of the world has almost no diversity in it. And that diversity, and we'll get to that in a second. But we'll see here kind of at the right side of this tree, we have these longer branches and much less closely related than the ones on the bottom left. And so this indicates that they have considerably more diversity than what we see in the majority of samples. And it turns out that much like Brucella melitensis, this has a geographic kind of origin, and that that nearly all of these samples come from Africa. They come from cattle in Africa. And so we have amazing diversity of Brucella abortus in Africa. And throughout much of the rest of the world, we actually have very little diversity. And in many cases, although not all cases, particularly with, say, plant domestication, if you're trying to find the origin, typically the origin of various species is where they are most diverse. And as they spread out from those areas, they get less and less diverse. And we'll see additional support for an African origin in a second. And so this is just another way to show that phylogenetic tree. And so at the top of this tree are those groups from Africa. Again, we see these long branches and lots of diversity within those African samples. We see a second group here, which is group C. And so this lineage is almost entirely comprised of samples that came from Asia and the Middle East. And I realize that Middle East is part of Asia, but you get the idea. Throughout the entire continent, there's a bit less diversity than we see in Africa, but we need to sample better from there. And then the rest of these come nearly entirely from Western Europe, North America, and South America. And compared to the other three groups, there's almost no diversity to kind of speak of. And so those branches are all very short, strongly suggesting that this happened recently and that these animals were spread very quickly with, highly similar Brucella abortus in them. And so this is just showing you some details of the African clade, kind of pointing out that there's two of them, that they're particularly diverse. This is the slightly less diverse Middle East and Asian clade or group. And then finally, this is group D with all the samples in it. And this is like, I see Adam laughing because this is like no diversity, and we kind of throw up our hands at that when we see that. And so we can actually use a molecular clock. So I showed you those mutations, or that SNP, those mutational differences between different strains. We can actually use those mutations to estimate how fast this bacteria is evolving. And so if we use those and put them into a molecular clock to estimate roughly the timing of these different events, it suggests that Brucella abortus as a species emerged roughly 5,400 years ago. So relatively recently, and more recently kind of than animal domestication. And I'd say the exact date, is less important, except that, it happened relatively recently in time. And we can actually date some of the different branches that we saw previously. And so we see here on the top, we see that these are the oldest of the lineages that we have, roughly about the time of Christ, perhaps a little bit earlier. But again, we need much more sampling to be anything definitive there. We see this group C, which has moved places in this tree, but you get the same idea. This is the Brucella abortus, then emerged in the Middle East and Asia, slightly after that. And then this most recent one, the youngest lineages are occurring in Europe and the Americas, and it happened relatively recently. Again, roughly about 1500. I wouldn't read too much into that, except to indicate that this expansion happened relatively recently in time, and it happened quickly across much of the world, at least the Western Hemisphere. And so one thing that I'll point out, and I found this figure kind of fascinating, nearly all the samples that we have, and so this is an analysis of cattle, not of Brucella, but of cattle, looking at genetic diversity in cattle. And so this is kind of the broad picture of what we see. And so we see that much of the world, at least parts of Asia, Western Europe, and North and South America have one group. And then the rest of the world has two other groups. We have a black group that's largely in Africa, and then we have a green group that's kind of mixed between India and parts of Asia, South America, and Africa. So it turns out nearly all the samples that we have, especially from that group D that had very little diversity, come from blue cattle. And so there's a direct connection, at least in my mind, between the spread of a few groups of cattle or a few kind of strains of cattle globally. And when they brought them, they brought that particular group of Brucella abortus with them. And so it turns out whenever we sample from the cattle that are in green or in black, we find new lineage, we find new diversity that we hadn't seen before, and that we've just not sampled well. We've sampled conveniently, and that turns out to be kind of in much of the industrialized world. And we've kind of avoided or haven't sampled effectively from other parts of the world. And so this is the kind of summary for that part of it. And so apparent African origin of the species indicated by both the timing on the phylogenetic tree and the substantial diversity that we see there. Then Brucella abortus emerged in the Middle East and Asia. We had a late emergence, relatively late emergence in Europe and North and South America that happened relatively rapidly. And the diversity in some regions of the world of Brucella suggests that we're not sampling that well, and we're missing hosts, or we're just missing, we're missing hosts and we're missing sampling events. And finally, Brucella abortus as a species is younger than previously estimated and occurred well after cattle domestication. And so I'm particularly interested in looking for new variation in parts of Brucella abortus variation in parts of Africa and Asia, and in fact have 3 new projects looking at exactly that. And one potential idea is that it may be in wildlife rather than in cattle. And so certainly it's one place that we're looking, yet at the same time, we have substantial diversity and unsampled diversity in cattle as well. And so we know that there are amazing diversity of cattle throughout Africa and India as well. And it'd be very interesting to look for Brucella there. So the main thing here is that Brucella has hitchhiked globally along with its animal hosts. We can use genomics to track its spread over these ancient timescales. And some regions are particularly poorly sampled, and we really kind of need to get out there to better understand this disease. And I should have led off with that hundreds of millions of animals worldwide are infected with brucellosis. It's arguably the most important livestock disease globally. And so the effects of Brucella are particularly pronounced in the developing world. And so sampling there, yes, it kind of helps satisfy kind of a scientific curiosity, but at the same time, we are solving kind of one of the major livestock diseases by understanding how it's transmitted both over short scales and longer time periods. And so with that, I can take any questions.
Excellent. Thanks, Dr. Foster. We do have, relating to your last slide there, we have two questions, and that is, what is brucellosis in terms of its symptoms and its outcomes? This is a livestock disease, but can humans also get it?
So brucellosis is, so it's a bacterial disease that we're primarily getting from unpasteurized milk. And so it's not the only reason, but it's one of the main reasons we actually pasteurize milk. When you heat up milk to high temperatures, it kills the bacteria, kills a couple of other bacteria as well. But, and so we don't have it in the US with a few exceptions, and we can talk about that. And so the symptoms are, in humans, they are typically arthritis and pain in the joints. It can cause reproductive failure in humans. They're high fever. It's called an undulating fever, where you get high fever and then it disappears, and then a high fever again. And so it kind of spikes and drops and spikes and drops. And those are the two major symptoms, and it typically becomes chronic. And so people will be infected with this for decades and just be basically miserable. But a lot of people don't seek help. It turns out that that the last human estimate was 500,000 people a year get infected, but the true number is probably 5 million people get infected a year, and it's just severely underdiagnosed. And it's difficult to treat. In animals, the primary symptom is abortion. So there's good reason why it's called Brucella abortus. It causes abortions in lots of different animals, and that has a major effect on the livestock health and productivity. So that's the major thing that we're trying to kind of guard against.
Excellent. Thanks that's a good answer. That was a lot of info. The next question is, does every animal or person with Brucella become infected, or some folks or animals just carriers who can spread the disease to others?
Yeah, I don't know of Brucella of any carriers. You get infected or you don't, but you're not spreading it to others. It's highly infectious. It's probably the second most infectious bacteria out there after Coxiella burnetii. And so it only takes a couple cells to infect people. In fact, it's the #1 laboratory acquired infection. So people that are trying to identify what bacteria are infecting, people in the clinic trying to identify what has infected people, infect themselves with it because it's so infectious. And so, but people can't spread it to each other. There's a few cases of kind of sexually transmitted diseases, but animals spread it to each other, but people don't.
Okay. The other question related to that is, if a pregnant female has Brucella, can the baby get it?
Yes. So both things happen. So usually there's an abortion. And so the bacteria love to live and they thrive in reproductive tissue. And so when that happens, they multiply into the billions, if not trillions, and that often causes abortion. But the key thing about that is that it's the way for the bacteria to get out and infect. And so that's their way of escaping the host and infecting other hosts, is coming out through abortions. But it does happen that some babies will survive, and typically they are infected and will be infected for life unless they are treated.
And that leads me to another question. What is a treatment for Brucella?
Yeah, so there aren't effective ones in animals. Typically, animals are killed because it's very difficult to get rid of it in animals. Although there are potential treatments, it is a 6 to 8 week, sometimes longer treatment with antibiotics. And so it's a combination of 2 different antibiotics that are typically given for up to 2 months. And it's very hard to kill because it's intracellular. It gets inside of cells, it gets inside of immune cells and hides out in those immune cells. And so when the immune system looks for it, it can't find it because it's in the cells that are sent to attack it.
Got it. Thanks. Sorry, my dog's losing her mind out front. Folks, we have plenty of time for more questions, so make sure you submit them. I'm going to ask a methodological question because I also work in phylogenetics, although I wish my fossils had DNA, but they don't. I was wondering, one is, what are you using as an outgroup in terms of non-Brucella? How are you rooting your trees? What non-Brucella but Brucella-like bacterium are you using to help, orient which direction sort of the mutations are heading?
Yeah.
And then the other is, if the B. abortus lineage has really mixed diversity, is that kind of screwing with your molecular clock in terms of mutation rates?
Yes, I'll answer the second one first. Yeah, so you may have seen, so there are 2 lineages within Africa. There's an A1 and a B1, and the B1 was much better resolved. And so, yes, certainly we want more data to have kind of convincing evidence of the dating of those dates for sure. And so, yes, I take any molecular dating with a grain of salt, but particularly in one of those lineages where we really want better sampling. And so, yes, kind of a great point. And so the other one was, remind me again.
Oh, just what are you rooting your trees on using an outgroup?
Yeah, so it turns out Brucella emerged from the soil, and I could have given a talk about that. And so all its close relatives are soil bacteria, and it somehow it's lost that ability, and we can actually see it in its genome. It doesn't have many of the genes that would allow it to survive in the environment, and so it has most of those genes have either been lost or they've been adapted for particular hosts. And so we can actually look, and there are a whole bunch of soil bacteria that are closely related enough that we can use them as outgroups and then kind of polarize or orient our trees. And so, we've recently found kind of some intermediate species where they are partially they can live in the soil, but they're not very good. And they have some of the genes of the soil bacteria, the Ochobacterium, and they have some of the gene loss of the Brucella, and they can live in particular hosts. And these were just found, and we're still trying to kind of make sense of all this new diversity, kind of a mix of both. And so, yeah, great question. So closely related soil bacteria.
Awesome. We have one more from the attendees, and that is, can Brucella spread to any animals like a predator?
Yeah, so yeah, I didn't talk about this. I have a bunch of slides for it, but we actually did a study in Yellowstone. And so it turns out one of the only places in the US where there's active brucellosis is in the greater Yellowstone region. So Idaho, Montana, and Wyoming. And it's still there today in bison and elk, and they spread it to cattle in the region. And so it turns out there have been some studies that haven't been actually haven't told us that much about Brucella in that system, but it looks like in coyotes and wolves. And so they've looked for Brucella in coyotes and wolves and haven't found it yet. But it turns out there's a Brucella that's found in dogs. It's called Brucella canis. We've actually had several cases of Brucella canis in dogs in Flagstaff recently. And so it's around. And it turns out that group from dogs is all the relatives of that are in pigs. And so it turns out it's almost certain that dogs got it from pigs, and then it evolved to just be within dogs themselves, or canids. And so like wolves and coyotes and other kind of dog-like animals. So yes, so predators can get infected for sure, but it doesn't look like they transmit it much beyond that, with the exception of the one that's specific to dogs.
Awesome. Well, if anybody else has any questions, feel free to put them in the chat. We're going to wrap it up here by just again reminding folks that we have a brand new app on Android, and Apple is the other one. We're really happy with it, and it's the first year for it. So make sure you check that out if you're interested. Oh, Dr. Foster, you got one more question. We can circle back.
So the question is, do javelina carry or transmit Brucella? And we haven't found it yet. I would highly suspect that they do, but yeah, we haven't found it.
Yeah,
so
go ahead, sorry.
Yeah, so the other question from Isabella, can Brucella affect the DNA of any organism? So for a long time, we thought Brucella only affected livestock. And then we found it in wood rats, in fact, and then we found it in caribou. And then we found it in marine mammals, and that blew our minds. And now wherever we look, we are finding it. We thought it was only in warm-blooded animals. We thought it was only in mammals. And then it's in amphibians, it's in snakes, it's in fish. It's wherever we look. And so probably not any organism, but it certainly seems like they can infect a broad range of vertebrates and potentially invertebrates. But yeah, the more we look, the more we find it. And for example, it's been found, maybe 5 different species of Brucella have been found in frogs in the last 2 years. Crazy differences that are kind of blowing our minds about what Brucella is and what it can infect. Yeah, wow. Great question. Thanks.
Well, I think that wraps up our Q&As. If you wouldn't mind relinquishing the screen, I'm going to show our sponsors real quick here. Remember how to do that. Oop, you don't need to see all of that. Can everybody see the sponsor slides here?
Yes.
Okay, so yeah. Oh, sorry, this is the survey. We are always interested in hearing what we did right, and sometimes more importantly, what we didn't do right with respect to not only this webinar, but any experience you've had with the Flagstaff Festival of Science this year. I've been asked to plug that anytime you put in the survey with your email address, you can potentially win a pair of AirPods. So there you go. And then, of course, as I reminded folks, this is going on now 3+ decades of a completely free Festival of Science here in Flagstaff. It's actually the world's oldest free Festival of Science. And we couldn't do it without our sponsors, but also the individual donors that we have that aren't listed on this. And if you are interested in sponsoring or donating, you can go to scifest.org because it brings to you great talks like this one and others. And we want to thank Dr. Foster for being here. And with that, we're going to wrap up. And again, thanks for coming. And everybody have a good night.
Thanks, everyone.